Okay, so today we are going to get started thinking about blood. We're definitely not going to finish all our blood stuff by the exam, so wherever we stop is where we stop. But I'll let you know about that as we move forward. A question for you all first, because I was not involved in physiology lab this fall. Have you guys seen this picture before? Did you do this in phys? Can people raise their hand if the answer is yes? Did they change this lab? Okay, I thought that might be the case. Okay, so in that case, if this does not look familiar from lab, we'll cover that in a little bit more depth and detail than I would otherwise. Okay. So as we're starting to talk about the blood, we're going to begin by thinking about some of the parts of the blood. And then we're going to move through some of the different types of blood cells. In class, we'll focus on red blood cells because that's going to be important as we move forward into our respiratory system. There's a little worksheet going through immune cells that I posted a link to, so we'll talk about that later. I don't really think for time we're going to get to that today, but we might. And then we'll move into clotting as our final topic. I suspect that will come after our next exam, based on my discussions with how far Dr. Schlater's class got today. So stay tuned. Okay, but we're thinking about the blood and we're thinking about its parts. Okay, so this first part, this question about plasma, you don't need some mysterious physiology lab to understand, because when we're thinking about the plasma, we should already know that the plasma is mainly made of water. Okay, so we're going to be overviewing the composition of blood, thinking about the plasma, erythrocytes, which are red blood cells, leukocytes are white blood cells, and thinking about platelets and hemostasis, which is like homeostasis, but for blood. And then some little clinical correlations at the end. Okay, so for our first question, all right, what the plasma is made of. We've been talking, whether you realize it or not, a fair bit about plasma as we've been thinking about fluid movement across our capillary walls, right? What we were really talking about was the fluid in the plasma, which is one of our forms of extracellular fluid. So we're thinking about the water in that plasma. That's the biggest component of the plasma, which is why when we talk about regulation of mean arterial pressure, we were saying that long-term regulation is going to be by the kidneys because we're regulating volume. That's going to have to do with that water component of the blood. So the next piece, we'll take a look at that little vial of blood and break down what the different parts are. But we have a measurement called the hematocrit. And this is telling us the percentage of erythrocytes by volume in blood. So basically, it's the percentage of blood that is made up of red blood cells. So that's going to be important when you're thinking about like how much oxygen can this blood carry? Because those red blood cells, those erythrocytes, their job is to carry oxygen. Okay. Finally, as we were thinking about that net filtration pressure, we were thinking about the osmotic oncotic pressures in the plasma, right? That was one of our pressures that we added together, one of our Starling's forces to figure out whether we have filtration, whether we have absorption. So this is just a fun fact that the biggest, not biggest by size, but like most of the proteins in your blood, the most prevalent one, let's say, is something called albumin. So if you're going to know the name of a protein, that would be a decent one to know. It's important in a clinical setting because it helps carry along a bunch of small molecules in your blood, including medicine. It's made by your liver. These are facts that were not originally in the slides. I just hate naming proteins and then not giving you any information about what they do, right? Drives me crazy to not know stuff. I guess that's how you end up a professor, but you know, side note. Okay, So we're thinking about the composition of blood. So we have a little blood vial over on the side. So this is what it would look like if you take a blood draw and then kind of run it through a centrifuge and sort of separate out its parts. Okay. So on average, blood volume for your textbook woman is about 5 liters, textbook man about 5.5, right? Almost certainly not very many of us would be exactly at the average, but that's approximately the range we're thinking about. Okay, and so we're breaking down our little blood vial, looking at what percent of that blood is made of what. Okay, so for your whole blood volume, that would be like your whole vial. Over half of it is made of plasma, right? So more than half of your blood is just going to be this clear-ish yellow part, plasma, which is mainly made up of water, right? So plasma is mainly water here, okay? Our other big component, right, I'm going to jump down to the bottom, are your erythrocytes. Your erythrocytes, so this part in red here. So those are your red blood cells. And that's what you would be looking for when you take a measurement of the hematocrit. So in men, typical values for the hematocrit are like 42 to 52%. Women a little lower, 37 to 47%. So that's saying what percent of this vial of blood is made up of those red blood cells. Okay. And then we have something called the buffy coat in the center. So this is just this little white layer between the two. So less than 1% of blood volume, but it's got some important stuff in it. Specifically, leukocytes are white blood cells, and platelets, which are almost like little fragments of cells that are really important for clotting, right? So it's not a big percentage, but important function here in the cells we find in our buffy coat. Okay. So here we have an overview of the production of our different types of blood cells and platelets. I'm going to walk you through. It's in the text later, but I like big pictures. Okay. So when we're thinking about all these cells, we're going to start from a single type of stem cell. So this is a hematopoietic stem cell, right? And basically, what we're seeing in this diagram is that hematopoietic stem cell can divide and specialize and become a lot of different types of blood cells. So we don't have to memorize all the different types of divisions. We'll be looking at important characteristics of different cells, but we can see that we have a split into 2 different types of stem cells here. So we have myeloid and lymphoid stem cells. You don't need to know those specific words. But then we're going to have further and further splits until we have all sorts of different types of leukocytes, or white blood cells. We have a lymphocyte over here. We also have little platelets. So you can see why I said that they kind of seem like little fragments of cells. I was watching a TikTok the other day that said that if they were foods, they thought they would taste like potato chips. And now that's all I can think of when I look at these little platelets, because they totally look like it. Sorry, again, an aside. And then we have our erythrocytes, our red blood cells, which we'll spend a lot of time talking about in class. Okay, but they're all coming from the same original hematopoietic stem cell. Okay. But back to our blood components. So when we're thinking about our plasma, it's mostly water. So it's like 90% water. We've got some proteins swimming around in there, 6 to 8%. And then we have our electrolytes. And by electrolytes, really, we mean salts, we mean ions, right? We talked a lot in A&P 1 about different ions and what they're doing. We will talk again about different ions in different systems as we move forward in this course, especially when we get to the kidneys, our renal system, okay? So we know we have especially lots of sodium, right? We talked a ton about sodium ions, especially when we were talking about the action potential in the nervous system. Chloride ions are also pretty high, right? So NaCl, it's like table salt, right? That's a salt, they stick together. In water, they split apart and form these ions. We also have some other important ions at lower concentrations. So we have hydrogen ions, which are basically just protons. We have bicarbonate in there. We have potassium ions. We have calcium. They all have their own role to play. So these are all dissolved into the plasma along with those proteins that are mixed in and really a lot of water. So our erythrocytes are what we're going to talk a lot about in this course. So these are our red blood cells. You have about 5 billion per milliliter of blood, tons and tons and tons of erythrocytes. They're pretty small, okay? So this is pictures of those erythrocytes. So we can see some properties of them already, right? So we describe them as a biconcave disc. So you're thinking like a donut, but the hole doesn't go all the way through, right? Concave is just referring to that shape. So concave, biconcave just means on two sides, and it's a disc because it's kind of flat. So if I were to slice across like this, actually, if I were to slice like that, right, that's what we might see, okay? Now this shape might not be obviously important to you, but it actually does really help with the function of the red blood cells. Because what this shape does is it makes it so that there's tons and tons of surface area on this erythrocyte, right? So everywhere I drew a line, right, that's the surface versus the inside, right? We have a really large surface area compared to the size of this erythrocyte, which means we're making it easy for things to diffuse in and out of the erythrocyte. So as we think about that, we're ultimately going to be talking about our red blood cells, our erythrocytes, as carrying gases, especially oxygen. So you can see why this might be a useful property for our erythrocytes to have, right? You want to load them up full of these gases quickly. You want to offload those gases quickly. So you don't want to have them needing to move really far to get in and out of this red blood cell. So that's why we care about their shape. So in diameter, we got about 8 microns across, thickness about 2 microns, okay? So those are just their measurements, right? Like that and like that. So they're small. Their membrane is flexible, so it's movable. It can squish around a bit. A special property of the erythrocytes, which distinguishes them from the rest of our blood cells, is that they don't have a nucleus and they don't have organelles. So when we think about the formation of an erythrocyte, they start out splitting from that hematopoietic stem cell. They develop all their different proteins. Some of those precursor cells, right, have organelles, have a nucleus so that they can make the proteins. But by the time you get a mature erythrocyte, those like die off, right? We hit delete, delete, delete on all our mitochondria, our nucleus, all those structures so that we can have this really small, really specialized cell. Okay. So this is great for their function. And so they can move along in your capillaries. They're small enough. They have lots of surface area for diffusion. But it means that they don't have mitochondria, means they are not going to be able to do that type of metabolism, that typical cellular respiration where we transport our pyruvate into the mitochondria, kick off that Krebs cycle, do the electron transport chain, the oxidative phosphorylation. They can't do any of that. There's no mitochondria, right? So they are not going to be able to get energy very easily because of this lack, okay? So their overall function is really just to transport oxygen and carbon dioxide. They can't really do much else because they don't really have many other cellular components inside them. If you are seeing red blood cells swimming around in like a patient in their blood and they do have organelles and a nucleus, that means you're actually getting some of the like earlier precursor cells migrating into the blood too soon. That's kind of a problem actually. So we're going to be talking about the life cycle of our erythrocytes because we're going to need to replace them because they can't really fix themselves. They can't do much metabolism. So we're going to need to make them up on a regular basis. But first, we're going to zoom in and think a little bit about that transport of gases. Okay. So inside our erythrocytes, the main thing we have are hemoglobin molecules. Okay. So that's what we really focus on when we think about red blood cells. We're thinking about hemoglobin because we're really caring about transport mostly of oxygen. Okay, so hemoglobin is like a globular protein in your blood. When you see this root heme, think blood, okay? right? So it's made up of 4 globin groups. So like polypeptide chains that folded and folded and folded, 4 of them. And we have our 4 subunits coming together. And then the heme group is in the center of each. That's this little flower looking thing. Okay. Okay. So we have the 4 globin parts, and then we have our little heme parts inside. Okay. So our hemoglobin molecule is really important for carrying oxygen. We can have a little bit of oxygen just dissolved in the plasma, right? So dissolved in that watery portion of the blood, but that's actually less than 2% of the oxygen that's traveling around in your body. Almost all of it, right? 98.5% of it is bound into this hemoglobin molecule. So this is why it's important to have enough red blood cells, why it's important to have enough hemoglobin. Okay. So when we look at these, we're going to see that the oxygen is going to be binding into that heme group, right? So that we have a zoomed in heme group. So heme, a notable thing to note about it is that it has iron in the center. So when you talk about like making sure to eat enough iron so you don't get anemia, right? This is why you need that iron. You need that iron so that you have it in the center of your heme group so that you can carry oxygen in your blood. Okay. So our oxygen is largely going to be binding into these heme groups inside our hemoglobin molecule. And in our respiratory unit, we'll talk extensively about how we load and unload that oxygen. What we want to know for now, when you're looking at someone's blood, you will note that oxygenated hemoglobin, also HB is short for hemoglobin. Okay, so that's just hemoglobin. Oxygenated hemoglobin is bright red, so that's what you'd see more in an artery, right? Deoxygenated hemoglobin is dark red, so that's kind of why we color the veins blue. It's because of that darker color, right? We're actually trying to show that different state of the blood. And you might actually see this, you know, if someone's drawing blood, if they draw it from the wrong place, you'll notice it's the wrong color, right? People are supposed to be drawing blood, I think, largely from veins, right? But if you get really, really bright red blood, it means someone probably hit an artery. Okay. All right. So one thing to note about the hemoglobin, just additionally here, we're going to talk tons and tons about hemoglobin and oxygen and how those things are related. But you should know that hemoglobin does also bind other things. So carbon dioxide, hydrogen ions, and carbon monoxide all do bind to hemoglobin. Not usually to the same place as the oxygen. Carbon monoxide is a special case. But when we think about carbon dioxide, it binds to hemoglobin, but it's not like competing with oxygen. It's binding into like other parts of like the globin part rather than to the heme part. Okay. So another thing that we see in these red blood cells is something called carbonic anhydrase. Okay, so carbonic anhydrase, right? It's going to be an enzyme. We know that because of this ACE ending, right? So it's going to help us transport gases, and it's going to do that by catalyzing a reaction because that's what our enzymes do. So specifically what carbonic anhydrase does is it's helping us convert carbon dioxide and water to carbonic acid and back. Okay. And as we look at this equation in the future, we will be paying attention to even more parts of it, right? When we're thinking about carbonic anhydrase, we're just doing this little part that I circled, right? So switching us back and forth between carbon dioxide and water and carbonic acid. But as we move forward, we're going to start thinking about the pH of your blood. And the pH of your blood is going to be dependent on the concentration of hydrogen ions and bicarbonate ions in your blood. So it is also the case that when you get that carbonic acid, it dissociates into those two products. So the hydrogen ions and the bicarbonate. So when we start to talk about pH balance, we're going to be thinking a fair bit about carbon dioxide, because ultimately, right, we're taking carbon dioxide and water and turning it into hydrogen bicarbonate. So this is going to be a way in which we kind of ultimately going to be thinking about carbon dioxide as a biological acid in the blood. Okay. And we'll talk about what that means and the implications of that later on. Okay. So for now, we want to know carbonic anhydrase is in our red blood cells, and it's important for helping us maintain acid-base balance of the blood because it's important for transporting carbon dioxide. So right now we're at the sort of existence and basic level, but we'll focus in a lot more on what that means and why we care about it later on. Okay. We're going to talk about those erythrocytes again. Okay. So when we're thinking about their life cycle, we know they don't have a nucleus, they don't have mitochondria, in fact, they don't have any organelles, they don't have DNA, they don't have RNA. They can't do stuff, especially they can't divide, right? You cannot make a copy of yourself if you don't have a nucleus and DNA and RNA and all that good stuff, right? So we cannot take a red blood cell and just make copies of it and make 2 red blood cells, right? We're going to need to go back basically to that stem cell level, those hematopoietic stem cells, split them out and specialize them, okay? So that whole process is actually going to be happening in your bone marrow. So in the inside of your bones, the marrow, inside the red bone marrow is where you have the production of erythrocytes, which is going to be known as erythropoiesis, just saying making red blood cells. The lifespan of an individual erythrocyte, an individual red blood cell, is about 120 days. You are replacing about 200 billion of your red blood cells every single day, right? So it's 120 days long, but it's not like you replace all of them all in one day. You're constantly having this turnover of red blood cells, okay? So they're going to be produced in the red bone marrow, and they're going to end up filtered out by the spleen and a little bit by the liver. So we're going to be making them coming from the bones, and then we're going to be killing them and stripping them for parts, ultimately in the spleen and the liver. Okay, so that's kind of our job for today, is to understand how we're making these red blood cells and how we're destroying them, but keeping the parts that we need. Okay, so our erythrocytes are going to be ultimately triggered to grow and develop by something called erythropoietin. So that's like our signal for making these red blood cells, okay? So it's going to be secreted by your kidneys when your kidneys notice that there's not enough oxygen in your blood, okay? So if your kidneys, which are filtering blood, notice there's not enough oxygen in your blood, they're going to say, hey, we need to carry more oxygen, and a great way to do that is to have more red blood cells, because red blood cells are full of hemoglobin. Hemoglobin carries oxygen. Okay? So under those conditions, the kidneys produce this chemical erythropoietin. This has a sort of flow of signaling that it kicks off. So hematopoietic growth factor is also involved in this process. So you're going to make that in response to the erythropoietin. So ultimately what we're doing, kidneys giving off this erythropoietin because your blood doesn't have enough oxygen, we're going to be triggering those stem cells to divide and become erythrocytes. Because when they divide, they could become a lot of different cells. That's what that big map at the beginning was supposed to show you. But we want these to now become erythrocytes because we want to carry more oxygen because we don't have enough oxygen in the blood. Okay. So as they develop, first, they are going to produce their hemoglobin, and hemoglobin is a protein. So they have to do that before they're fully mature erythrocytes, because erythrocytes, red blood cells, don't have DNA, RNA, et cetera. So they actually can't make proteins, right? They don't have the machinery. So throughout this process, as they're developing, they're going to be making that hemoglobin. And then ultimately, when they're ready, they're going to lose their nuclei and organelles so they can be nice and small, nice and agile, have that good surface area, allow for lots of gas exchange. Okay. Now we have some requirements for the production of our erythrocytes. Right? You need specific stuff in order to make a red blood cell. The main one that we think about on like a day-to-day basis in our own lives is probably iron. So iron is in that heme portion of the hemoglobin. Right? So you need iron in the blood in order to make effective hemoglobin and therefore to produce good erythrocytes. So the normal hemoglobin content of blood is somewhere around, let's say, 15 grams per deciliter. That's me eyeballing. What's a good number? Kind of as an average in there. But there's a specific range you would expect, somewhere in those ranges. Okay. Now you also need folic acid. So folic acid is actually something that you need for DNA replication. So anytime you want to make lots of cells, you're going to want folic acid. So this is why also in like pregnancy, early pregnancy, folic acid is a supplement that people take because you need to do a lot of DNA replication and cell proliferation if you're trying to grow a fetus, right? So that's kind of a connection there between things you might see in vitamins. Okay. The other thing is vitamin B12. You also need that for DNA replication and cell proliferation. So anytime you're trying to grow new cells, new tissues, you're going to want folic acid and vitamin B12. So you need them for your erythrocytes, just like everything else. And you need that iron because you're trying to make blood. You're trying to make hemoglobin. It needs that iron to carry the oxygen. All right. So those are the things we need. So we're going to make our red blood cells. And then at the end, we're also going to filter and destroy them. Okay. So the spleen is going to be filtering blood and pulling out the old crusty looking erythrocytes, right? The ones that are starting to break down, don't look so functional. Spleen's pulling them out and removing them. And then your liver is helping you break them down and cannibalize them for parts, right? So your liver is going to help you kind of metabolize the byproducts that you're producing as you break down those red blood cells. Okay, so the spleen has these cells called macrophages in them. What the macrophages do is they are filtering the blood and they are basically eating up old fragile red blood cells. That's what phagocytosis means. They're encircling them, pulling them in and breaking them down. So that's what these macrophages do. Okay. So they're pulling in those red blood cells, they're breaking them down. Specifically, we're going to be breaking down the hemoglobin. So catabolism is making something smaller, breaking something up. So when we see that hemoglobin is catabolized, we're saying we're breaking apart that hemoglobin. Yeah. So the hemoglobin had those 4 globin subunits and then those heme groups inside of each. And we're going to deal with some of those parts of that molecule kind of separately. They're going to become different things in your blood. So once you remove the iron, heme becomes something called bilirubin. Bilirubin then goes into your bloodstream where it gets transferred into the liver and further metabolized. Okay. And then we're going to excrete some of the stuff in bile that goes into your intestinal tract. And this is actually what gives feces that color, is partially from the bile coming from this bilirubin. And other parts of this are going to get released into the bloodstream and excreted in the urine. I know I'm going a little fast. That's because I want to get to the picture because I like talking while looking at pictures. All right. So when we talk about the iron, most of it we're going to want to keep, right? We don't want to just like get rid of all your iron, excrete it out. We actually want to recycle it so that you don't have to constantly be eating little bits of metal, right? So you're going to recycle that iron for synthesis of new hemoglobin, which means that we're going to have to transport that iron around in your blood as well. So some of the iron you're transporting is from stuff you're eating, but a fair portion of that iron transporting in your blood is coming from old hemoglobin that you broke apart, and you're carrying that iron around so you can reuse it. Okay. So we're going to see iron moving around in the blood bound to a molecule called transferrin. So in the figure we're going to see, I think that's going to be labeled T iron in a fair number of places. All that means is going to be iron bound to transferrin. Transferrin is just transferring the iron, okay? And actually, it tells you in that as well. Ferrin, that fer root, it refers to iron. So we're transferring ferrin. Ferrous is iron. Okay. So that transferrin is going to be moving our iron from the GI tract. So your gastrointestinal tract, your intestines, where you're processing food that you've just eaten. We're going to take some of that iron from the GI tract, ultimately move it into the bone marrow. We're also moving iron from the liver into the bone marrow because the liver is going to be part of this whole processing system for the breaking down of red blood cells. When we store iron for use for later, if we're not actively right then making a bunch of red blood cells, if we have extra, we're going to bind it to something called ferritin. So when you see F iron, that's just telling you it's in this sort of storage form. Okay. So ferritin is something that helps us store iron. We're going to see this in our liver, our spleen, and in the small intestine. Cool. So we're going to walk through this picture in pieces. Okay. So to kind of understand what's going on. Okay. Actually, I'm going to take us to the big picture real quick. So we're going to start with the birth of our red blood cells in the bone marrow. But I'm just showing you here that we do also on this image have our signal coming from our kidneys. So we're going to kind of be looping around here. Okay. All right. So right now we're looking at our cardiovascular system. That's our red tube. Right? And we're starting out with our bone marrow. Okay. So in our red bone marrow, so the marrow cavity is this hollow part inside, right? So we can see that our bones are hollow inside, right? They're filled with bone marrow. This is where we have those hematopoietic stem cells that are going to allow us to make erythrocytes, okay? So the erythropoiesis is happening up here in the bone marrow, creating our red blood cells, which we also call erythrocytes, which then migrate out of the bone and into the blood. Okay. They're moving bone marrow out of the bone and into the blood, essentially, these products of the bone marrow. All right. We are already getting towards the recycling portion, right? So during their lifetime, those erythrocytes are swimming around in your cardiovascular system. They are carrying gases back and forth to tissue, right? Oxygen bound into the heme groups and the hemoglobin, but also we got some carbon dioxide in there, some other sort of byproducts. But ultimately, when we hit that approximately 120-day mark, these erythrocytes, as they go through the spleen is going to say, hey, no, this one's kind of broken. We're done. Okay. So we're going to start to break apart that erythrocyte. So the erythrocyte here is traveling through your spleen. Okay. And we can see now that we're beginning to break it down. We're breaking apart the heme group from the globin group. So we had our big hemoglobin molecule, which was, right, like globin, 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 globin. Heme. Okay. All right. So we're breaking apart the heme from the globin. We're pulling those heme groups out. Okay. Now we can see now we can see the sort of fate of each of those products. Okay. So we're going to focus first on the heme. Let's start with. Okay. So the heme was that little yellow flower with the iron in the center. Okay. So that heme, we're pulling the iron out, and then we're going to transfer that iron because we're going to want to reuse it. So that's why we now see T iron in the blood. All right. So we took the iron out of the heme and moved it around in the blood bound to transferrin. Okay. Now, when we broke apart that heme group, we didn't just have iron. We had the rest of that little yellow flower thing. So part of that is becoming bilirubin. Okay, which is also going into the blood. And another part of the bilirubin is just all the bits from the globin part of the molecule. Okay, so the globin part of the molecule, we didn't zoom in on its structure, but like any other protein, right, it can be thought of as a bunch of amino acids stuck together, right, in a polypeptide chain that just folded a bunch, right? So that's like what the globin would look like, okay? So when we break down hemoglobin, we break out those amino acids, and they also become part of the bilirubin, okay? Now, for prioritizing here, we most want to understand what happens to the heme portion and the iron, because that's what we're going to be reusing in this cycle, right? So that's our most important for function in the body. All of everything your body does is kind of important, but this is going to be important to make sure that you can continue to have functional red blood cells carrying oxygen. Okay. So now we're going to see the liver. Okay. So this was our spleen in brown here. Now we're down to our liver. Okay. So we can see one thing that's happening in the liver is that we're taking that bilirubin, we're making bile, and then this is just a new form of waste product, essentially, is that bile. Okay. So that's one piece. So all of this would have been a flow, the globin to the amino acids to the bilirubin. Now we're in the liver, bile, we got bile, okay? I'm circling that as one group because the other thing happening here is kind of separate. So the other thing we see is the F iron to the T iron. So the F iron isn't coming from the bilirubin. The F iron was that storage form of the molecule, right? So the ferritin bound to iron. So what this is showing us here is that some of the iron in our blood was iron that we pulled out of a heme group and bound to transferrin. Okay. Some of the other iron in our blood, as we're getting ready to make more red blood cells, is coming from this storage form of iron, the F iron. And then that's also getting transported into the blood. Now we're binding it to transferrin. So that's why it says T iron, right? Because that's just the way we transfer iron around in the blood. All right. So now we see a lot more on this map. Okay. So we saw some of the bile going around in the bloodstream, but where you're mostly going to think about bile probably in your lives is when you think about the intestines. So bile is important for digestion, and you're also going to see in anatomy lab that it's like really bright green when you see it coming out of the liver and getting stored in the gallbladder. It's going to be pretty noticeable. Okay. It changes color as it moves through your digestive tract and then gives feces that brown color, right? So that bile, lots of it is traveling through your intestinal tract, allowing you to break down fats, but ultimately getting excreted, right? So that's kind of what's happening to the globin part of the hemoglobin, right? It was globin, then amino acids, bilirubin, bile. Now it's helping us break down fats and getting excreted. Okay. So that's one sort of pathway that's happening here in the intestines. And the other thing we have going on is our dietary iron. Okay. So your small intestine is the first stop for food you eat. So we'd be coming in through your mouth, your esophagus, your stomach, ending up in your small intestine. So you've eaten something with iron, an iron supplement. I don't know, maybe you cooked with cast iron. Now there's a little bits of iron in your food, wherever you got this iron, spinach, who knows? We're going to soak some of that up, move it into your blood. And since we're moving it around, it's got to be bound to the T iron. Okay. So these are our three sources of iron in the blood, right? We had iron coming from breakdown of that heme group, bound to transferrin. We had iron that was stored in the liver bound to ferritin that we can then move into the blood bound to T iron. And then iron that we eat also moves into the blood, moved around as T iron. Okay. All right. So we have this iron circulating around. We have our red blood cells circulating around. Right? And then we are going to see things filtering into the kidneys. So one of the things that is filtering into the kidneys is any bile that didn't go into the intestines gets excreted out as urine instead of feces. Right, so some of that bile is basically kind of dissolved in the blood plasma. Okay. But what I consider our more important role of the kidneys for this life cycle is the fact that as we're processing blood through the kidneys and determining how much blood volume we want, how much water we want in the blood, we can sense a little bit about the oxygenation content. So we're sensing whether we need more red blood cells. And if we do, we give off that erythropoietin, which is a signal to our stem cells in the bone marrow to divide and become erythrocytes. Okay. So the final little bit that we're adding on here that we didn't see before is that in order to do that, they're taking all of that T iron, right? So we saw one source of iron here from the heme, another source from the stored iron in the liver, a third source from your dietary iron. However it got there, it's in your blood. So your bone marrow is going to pull it in and use it to make new red blood cells. So that's kind of our whole process for this production and destruction of red blood cells. So questions about this process at this point? Yeah.
So like the F iron, like, just does it just stay basically in the liver after it gets there?
Yeah, so the F iron is like a storage form of the molecule. So like any excess iron can get stored in the liver until you want it later. Yeah. Does that make sense? Yeah. And I think like if anybody's eaten liver and onions, I swear you can like taste that iron, right? My dad's super into liver and onions. I think it's gross, but whatever. I've tried it. Questions about this life cycle? We're going to just do this one last slide. You know, we have 5 minutes left. We're not going to break into all of our other types of blood cells. So my only additional point for you here is understanding what anemia means, which is basically just that in your blood, for various different reasons, you can have a lower carrying capacity for oxygen, right? Your blood can have problems with it so that you can't carry enough oxygen. So that is what anemia means broadly, but we can get more specific and give different reasons for that lack of oxygen, right? Jumping down to the bottom of the list, right? You even have one that's just, you are bleeding, so you're not carrying enough oxygen, right? So hemorrhagic anemia is just like you're bleeding out, so you don't have enough blood, you don't have enough red blood cells, they're not carrying enough oxygen, right? So anemia is really just about that carrying capacity for oxygen and that hemoglobin. Okay. One of the most common ones that you'll see is dietary anemia. So if you go to the doctor and they tell you you're anemic, probably that's what's going on, right? They're going to tell you to eat more iron, maybe tell you to eat some more vitamins, right? So dietary anemia has to do with the fact that you're not eating enough of these basically precursor products that you need, right? So you need iron to make those heme groups. You need vitamin B12 for that cell division. So we might also call it pernicious anemia if the problem is that we're not able to make those new red blood cells because we don't have enough dietary products. Hemolytic anemia has to do with the properties of the blood cells themselves. One of the most common types you'll see is sickle cell anemia. So sickle cell anemia, which is a condition some people have, it's genetic. It has to do with the shape of your red blood cells, right? If you have this one slight mutation, instead of forming that typical hemoglobin structure with those 4 globin subunits, you can end up with hemoglobin that kind of crystallizes in a sort of sickle shape, and that actually affects the shape of the red blood cells. And that has pros and cons. The con is you're not going to carry enough oxygen potentially. And these red blood cells can kind of get stuck almost in capillaries because they're a different shape. But the plus side is that it defends you against malaria, right? So that's part of why we do see this condition, is that in some areas it's been adaptive in human history. Okay. We also have types of anemia that are coming from other systems. So when we talk about aplastic anemia, this has to do with issues with bone marrow, which now that we know that our red blood cells are made in the bone marrow, makes sense. Your bone marrow has a defect. You can't really make enough of these red blood cells. Okay. And then finally, renal anemia. If you have kidney disease, we know that those kidneys are what's sending the signal to make our red blood cells, right? So if you have certain types of kidney disease, renal anemia can be a result of that, as you're not making enough of these red blood cells or in the right way. And therefore you don't have this carrying capacity of oxygen in those red blood cells. Okay, so that's where we're going to end for today. So Friday we'll be reviewing for our exam on Monday. Very sorry that we didn't look at the Super Bowl schedule when we made the schedule for the exam. I'm sorry. Wasn't on my mind.